This unit is on cell division. Cell division is how we grow from a single cell to a full-grown adult. So you may ask yourself, why do cells not just keep growing to fill a bunch of space? So cells divide for a very specific purpose. That purpose is to maintain what we call a surface area to volume ratio. Cells cannot function very well if they are too big. So here's an example for you. I'll kind of explain this diagram that you're looking at. So in this picture, we have a cube that is 5 by 5. Okay, length, width, height, all 5 by 5 because it's a cube. All sides are even. That has a surface area of 150. The units are irrelevant, but that has a surface area of 150. The volume is 125 based on how we figure out volume. So our ratio is 1.2, surface area to volume. Well, if we take that same cube and cut it up into bunches of little smaller cubes, we increase the surface area to 750. Now think about it this way. If you took one piece of toast and your goal is to soak up as much honey with that piece of toast as humanly possible, you would like to cut the toast up into lots of small pieces so that more of the toast can interact with the honey. Think about all the sides that are created when you cut the toast up into different pieces rather than eating the whole big slice. It only holds so much honey. So that's why cells divide. If a cell is too big, it cannot efficiently transport materials from the outside of the cell wall, to the inside of the cell, to the nucleus. Think about it in a big, huge room. It takes more steps to get from the door to the middle of the room if the room is really big than if you cut that room into lots of smaller chunks. In the door to the center of the space takes less energy, less steps. So the whole point is for the cell to be able to conserve energy to make it more efficient. Okay, so the cell cycle is the life cycle of a cell. It's divided into 5 main stages. Well, let's call it 3 stages. Okay, 3 main stages. The first stage is called interphase. Interphase has 3 parts, G1, S, and G2. In the first stage, gap 1, everything is duplicated or copied except for the chromosomes or DNA. S is when we do synthesis, which is DNA is duplicated. So we make copies of all of that important stuff that makes up who you are. The next part of interphase is gap 2, which is our spell check phase. So it's just like writing an essay. You have to have that double check point, make sure everything's right before you continue on through division. If something is terribly wrong in the cell and it cannot be repaired, the cell will initiate what we call apoptosis, which is kind of a fun word, but what it really means is cell suicide or cell death. So if the cell cannot correct everything that is wrong when it gets to G2, then it will kill itself. It will stop, and hopefully, if it's terribly wrong, it will not go on to continue through mitosis. The next step is mitosis. This is the word for nuclear division. Mitosis means nuclear division. And it is dividing the nucleus into two separate nuclei. Because if you're making a new cell, you have to have nucleus for one cell and a nucleus for another cell. And then the last step is called cytokinesis, or division of the cytoplasm and the organelles. So we made new organelles in gap 1. And so we have 2 copies of the mitochondria, 2 copies of the lysosomes, 2 copies of the chloroplast in our cell. Then we divide the DNA, then we double check everything, then we divide the nucleus, and then we divide everything else. So it's like a divorce. You've duplicated everything though, so it's not like you get the couch, you get the chair. Everything has been duplicated and evenly divided into the 2 new cells. So mitosis has 4 stages. So we don't really consider interphase a part of mitosis. It is that beginning stage that comes before mitosis because it has those 3 parts, G1, S, and G2. So in interphase, the cell more or less looks like this. Everything's been spell checked, but know that there are other organelles in this diagram here. You just cannot see them. They're not drawn for the sake of simplicity in these pictures. So the first stage of mitosis is called prophase, and it looks kind of like this. And we're going to go through what each of these stages means here in a second. The second is metaphase, the third is anaphase, and the fourth is telophase. A mnemonic to help you remember this is PMAT, prophase, metaphase, anaphase, telophase. So you should connect mitosis, PMAT, mitosis, PMAT. The first step, prophase, the first step of mitosis is called prophase. This is when chromosomes condense. We're going to get to in just a second what are chromosomes, but for now, in short, chromosomes are DNA. During prophase, that membrane that wraps around the nucleus, making it its own entity, starts to disappear. Your centrioles move to the poles. So if you remember way back to our cell booklet, centrioles are organelles that are used in the process of cell division. So in this, they move to the poles, literally to North Pole and the South Pole. They're just opposite each other. It could be east and west too, but they're opposite from each other, and we call those the poles of the cell. And then these spindle fibers form. So they kind of look like spider legs at this point. And you'll see their importance here in just a little bit. Okay, so I told you we would get to what is chromosomes. So there's a terminology difference I want you to pay close attention to here. Chromatin versus chromosomes. So you'll hear these words used somewhat interchangeably, but they are different and they have different meanings. So I want you to be sure you understand it. Chromatin is not condensed. Chromatin, not condensed, but it is a copy of DNA. So it is duplicated DNA. So you take one chromatin or chromatid and another chromatid and you put them together and they're not condensed, and that is chromatin. So I like to think of chromatin like a spaghetti noodle. It would be found in the nucleus like a cooked piece of spaghetti on a plate. And then chromosomes are condensed DNA. They are wrapped up like you would wrap spaghetti around a fork before you get ready to eat it. That's what chromosomes are. They're condensed, wrapped up around proteins so that we can have them in nice compact form so that when we finish mitosis that every cell ends up identical to the original cell. So a chromosome is paired chromatids. So a chromatid is one piece of spaghetti. And they are identical to each other. The centromere, if you're looking at your picture here, is the blue dot that connects the 2 chromatids. It's right in the middle of the sister chromatids. All of this X shape is chromatin. wrap it around a fork, it's called a chromosome. So I know that terminology is a little bit confusing, but I have confidence that you can understand it. All right, the next step is called metaphase. Meta means middle in this context. So the chromosomes that we've condensed down in prophase line up across the middle of the cell, and those spindle fibers that were beginning to be produced in prophase are now stretched out, attaching to the centromere of each chromosome. So it attaches right to the middle of the chromosome, which will be very important later on. But just so you remember, each side of this X-shaped chromosome is identical to each other. So metaphase, the chromosomes are across the middle of the cell. The next step, anaphase. This is when the sister chromatids separate or move apart. Ana, apart. So A and A. In this phase, the way it happens is the spindle fibers shorten. So think about you've taken a rope, spindle fiber, lassoed the centromere of the chromosome. Now, how do you get the lasso in? You pull it back. You shorten your fiber, which then brings that chromosome towards the pole. So here you can see in your picture, you have centrioles on either end of the cell. You have spindle fibers attached at the centromere of the DNA of the chromosome, and they're shortening. They're pulling in those chromosomes to the opposite ends of the cell. The last step of mitosis, the T in PMAT, is telophase, and it means that we now have 2 new nuclei. So if you've noticed, with every letter we have kind of a way of remembering it. So we have PMAT, prophase is when they condense, metaphase, middle, meta, middle, anaphase, apart, telophase is 2. So we kind of go along with the letters of PMAT for every single stage. But in telophase, what happens is we have 2 new nuclei, and the nucleus starts to wrap back around its DNA. That's the job of the nucleus, is to maintain that DNA in a nice, neat little package. So during telophase, the chromosome starts to unwrap again, go back to its original state of chromatin. But remember, at this point, it's really more like chromatid because it's not a duplicated strand. It's just a single in its new cell. And so we have chromosomes uncoiling, we have that nuclear membrane wrapping back around the DNA, and all those spindle fibers that we used in anaphase are falling apart, and their components are being used in other parts of the cell cycle. Okay, the last step of the cell cycle is called cytokinesis. So cytokinesis is the completion of cell division. It's the finishing product of cell division. This is when we have the cytoplasm and the organelles being divided up between the 2 new cells, and the cell membrane or the cell wall starts to reform. So you know, in a plant cell, you have a cell wall and a cell membrane. So the way that cytokinesis has to happen in a plant cell is different than how it has to happen in an animal cell, because it has to build a wall back. So if you think about a plant cell, this is the picture of the plant cell over here in the green. What happens is this cell plate starts to form, and it's basically like building a brick wall back up between these 2 new plant cells. So they've divided, they've done all of the PMAT process, and now they're building a new wall between the 2 new cells. And that wall has a cell membrane and a cell wall. The animal cell is a little bit different. It uses the cleavage furrow. So I think a funny analogy to help you remember the cleavage furrow is, if you've seen the video on YouTube that's going around of the people that cut the watermelon in half by wrapping rubber bands on it, just putting as many rubber bands until the watermelon just explodes and is cut completely in half. Well, that's kind of like the cleavage furrow. It's like a whole bunch of rubber bands being wrapped around the cells until ultimately the cells break apart from each other. They're literally being pinched in half at this cleavage furrow. So we're going to switch gears for just a second and talk about the cell cycle and how it's regulated. So there are lots of steps to the cell cycle, and controlling that cell cycle is crucial to healthy functioning of a human, healthy functioning of all organisms. So the way that we control the cell cycle is through some signals. There are internal signals that come from within the cell, things like enzymes that are produced inside of the cell that control whether it's dividing or not dividing. And then we have signals from the outside that help determine if the cell is going to continue through the cell cycle. So external signals, internal, that's kind of self-explanatory. It comes from within the cell. But external signals can be a number of things. They can be physical signals from the environment. Like if you're in a crowded space and everyone is touching you, that would be a signal not to add any more people to the space. So that would be an external signal. If cells are too closely packed, cell division is going to stop happening in those cells. If cells can tell that there's nothing else around them, they will continue to divide until they are touching other cells so that they can share resources and things like that. But they can respond to a variety of physical signals from their environment. Cells can respond to hormones. Those hormones tell the cells to divide or to not divide. Those are all produced by other cells but received by a cell to say, okay, I'll keep dividing or I won't keep dividing. And some signals come from within the cell. A checkpoint in the cell cycle is a control point where stop and go regulate the cycle. So I have a diagram that helps to explain a little bit more. So at all of the big points of the cell cycle, it's literally like there's a stoplight. So we go through interphase, we go from G1 to S to G2, but before the cell can pass into mitosis, it has this stoplight. If the stoplight is red, then the cell is not going to continue through nuclear division or mitosis. If the stoplight's green, the cell is going to go ahead and start through the process of PMAT. At the end of mitosis, the cell is going to have a very similar checkpoint. It's going to have a stop or a go. If it's a red light, it's not going to continue into cytokinesis until all of telophase is finished. If it's a green light, it's going to continue into cytokinesis and the cell will divide its cytoplasm. Perhaps the most important checkpoint, though, is this one that happens just after cytokinesis, and it determines whether the cell goes back through interphase again or into this process called G0. That means the cell stops dividing. G0 is it's just growing, maintaining status quo, not changing anything. So if the cell gets a red light at this checkpoint, whether it's from something on the outside, whether it's something in the inside of the cell, then it will enter into this cell cycle arrest, G0 stage. If it gets a green light, it's going to continue into interphase, go through mitosis again until it hits this stop sign here, this stoplight here, and gets a no more sign, okay? So this is where this is really important and really interesting, and you can make some real world connections. So this is important because of something that you all know called cancer. Cancer cells do not follow those normal signals. They are the cars that run the stoplight. So if the cell gives a signal or receives a signal from outside that says stop dividing. Cancer cells do not. Now, we don't know the cause of all of these things. We don't know exactly if it's an environmental signal or if it's a chemical signal. Whatever it may be, we're not sure for every type of cancers. There are some that we know that are triggers, but there are others that we're not entirely sure of. All we know is that cancer cells, they just say, to heck with that stoplight and continue to divide uncontrollably. Normally, if a cell bypasses, runs through a stoplight, the immune system says, oh, that's a rogue cell, and goes and destroys the cell. Okay? But if the cell continues to get away from the immune system, then it will divide, and ultimately those cells will become cancer cells, and there'll be a whole host of people a whole host of cells that don't stop at the stoplight. There are a couple of types of tumors that can develop inside of the body. The first is a malignant tumor, and it invades or impairs multiple different tissues or organs, and it is a malignant tumor, meaning it affects multiple things. The second is a benign tumor. It's abnormal cells that are in the original site, relatively harmless. Usually you do have benign tumors removed if they're discovered early enough and in the benign stage, because you don't want for them to become malignant. And there are a whole host of things that are benign tumors that we're not going to go into. But they're just abnormal cells. They're not hurting anything. They're just hanging out, continuing to divide. Now, cancer can spread. It can separate from the original tumor. So if a mass is malignant, it is impairing multiple organs, which means it has the potential to enter the blood and the lymph vessels. So lymph is just like the blood, just like your circulatory system. It travels throughout your entire body except for your immune system. So it can get into those systems and implant themselves any other place in the body. If cancer cells implant in other parts of the body, we call it metastasis. So the cancer is said to have metastasized or spread throughout the body. That's usually not something that you want to hear. So here's a diagram to help. So malignant tumors are invading multiple different types of tissue. So you see 3 different types of tissue here: pink, blue, and salmon. So a malignant tumor, this one is invading the pink and the blue, and here on the far right, it's invading the pink, the blue, the salmon, and the blood vessels. As opposed to this benign tumor, it's just a local uncontrolled cell division, local set of cells that are rogue, but they're not hurting anything. They're pretty harmless. Malignant tumors can enter the blood vessels and metastasize to different parts of the body. So that concludes your notes on cell division for now. We'll pick up when we talk about division of sex cells.